human cell is divided into two divisions known as the nucleus and cytoplasm. It is very common for people to compare the inside of a cell to a raw egg, whereas the nucleus resembles the yellow yolk within the egg and the cytoplasm resembles the white part of the egg. The outer layer of a cell is known as the cell membrane. These structures all serve their own purposes and they all work together to form and operate the cell. The nucleus is known as the power center of the cell. It contains all genetic information and has full control over protein synthesis. Surrounding the nucleus is a double-layered structure known as the nuclear membrane. This structure contains large pores that allow substances to move freely between the nucleus itself and the cytoplasm. The base of the nucleus is filled with a fluid-like substance known as nucleoplasm. This nucleoplasm substance is composed of nucleolus and chromatin. Nucleolus, also known as the little nucleus, synthesizes RNA and ribosomes as they pass through the nuclear pores en route to the cytoplasm. Chromatin is composed of DNA strands which carry all genetic codes. The cytoplasm division, or the gel in the cell, is actually located within the cell but outside of the nucleus. This division is composed of mainly cytosol and a variety of small organelles. Cytosol is an intracellular fluid that is mainly composed of waters, electrolytes, proteins, and nutrients. Within this fluid resides our small organelles who all serve their own purpose. The mitochondria serves as a power plant for the cell. Endoplasmic reticulum are membranes that create channels that promote the flow of protein. The Golgi apparatus finalizes the protein and stores it for export. Ribosomes are where protein synthesis occurs. Lysosomes are responsible for phagocytosis and also removing damaged organelles. The cytoskeleton promotes shape, support, and function. The centrioles help separate the chromosomes during mitosis, and the inclusion bodies serve as temporary glycogen granule and melanin. The cell membrane, also known as the cell case, separates intracellular material from extracellular material. Intracellular material resides on the inside of the cell, while extracellular material resides on the outside of the cell. This case physically holds the cell together and regulates the entry and exit of any substance. This act of regulation from the membrane makes it semi-permeable, composed of phospholipids, proteins, and a small amount of carbohydrates. The protein molecules within the membrane serves as structural support and also acts as a binding site for hormones. Operating on the surface of the membrane, there are several structures who all serve their own purpose. Microvilli generates folds on the surface of the membrane, which increases surface area, ultimately increasing the amount of fluids that are being absorbed. This function is associated with cells who are involved with the movement of water and its solutes. Cilia are short, thin projections on the outer surface of the membrane who are responsible for transporting substances across the surface of the cell using wave-like motions. This is the action of clearing respiratory passages. Similar to cilia, flagella, which is a bit wider and longer, is actually responsible for moving the cell itself with its tail or whip-like structure. A variety of mechanisms are utilized to help assist the movement of water and its solutes across the cell membrane. This action is vital as it ensures the cell receives all of its necessary nutrients. These mechanisms can be divided into two different categories known as passive transport and active transport. Passive transport mechanisms can be described as a car in neutral coming down an incline. It needs no additional push or energy. Let's talk about a few passive transport mechanisms. Diffusion is the transportation of a substance from a high concentration area to a low concentration area. Facilitated diffusion is when the helper molecule assists in the process of diffusion. Osmosis is the diffusion of water solvent from an area of more water to an area of less water. And filtration is the diffusion of water and dissolved substances from an area of high pressure to an area of low pressure. On the other hand, active transport mechanisms can be described as a car neutral attempting to go uphill. This would require an input of energy. Active transport pump moves substance uphill from an area of low concentration to an area of high concentration. This requires an input of energy from adenosine triphosphate. Endocytosis is the ingestion of a substance by the cell membrane. This can also be split into two categories known as phagocytosis, which is the engulfment of a solid substance, and panocytosis is the engulfing of liquid droplets. Exocytosis is the secretion of all cellular products out of the cell. The series of events that the cell undergoes from one mitotic division to the next is known as cell cycle. This cycle can be divided into two phases, interphase and mitosis. During interphase, the cell functions normally and prepares for mitosis through growth and DNA replication. Interphase is divided into three different phases. We have first gap phase, also known as G1, phase S, and we have second gap phase known as G2. During first gap phase, the cell tends to its normal activity and begins to make DNA and other substance necessary for cell division. During phase S, the cell duplicates its chromosomes, generating enough DNA for two identical cells. Second gap phase is the final preparatory phase for cell division and includes the synthesis of enzymes and proteins. Once G2 is complete, the cell enters the mitotic phase. During mitosis, one cell splits into two in a manner that the nuclei of both cells can retain identical genetic information. The mitotic division consists of four different phases: prophase, metaphase, anaphase, and telophase. During prophase of mitosis, the chromosomes coil tightly and are visible under a light microscope. These chromosomes are condensed of genetic material into two identical daughter cells. During metaphase of mitosis, chromatids are aligned to a narrow middle zone where spindle fibers connect them to centrioles. Anaphase occurs once the centromere splits, and it ends once chromatids all reach opposite poles. Telophase is when each new cell reverts to its interphase state. The nuclear membrane is reformed, chromatid strands start to reappear, and the chromosomes begin to uncoil. At the very end of the cell cycle, the daughter cells can either re-enter G1, where it will repeat the cycle once again, or it can enter a phase known as G0, which is a dropout phase.